Hello guys, my name is Dr. Varun and you are watching Chlorophyll classes only on YouTube. Well, this is another day for the 12th class. Of course, we are meeting after a really long time and we have to complete a lot of things this month. But it will be completed because I am there with you. Well, in this video, we're going to be starting a new chapter from your 12th class, that is molecular basis of inheritance. Molecular basis of inheritance. Till now, what we have studied? We have studied that there is what? Genetic variations and inheritance, means something related with the genes. But how that is actually working, how anything is actually coming up, this we're gonna be seeing over here, how it actually showing it effects. Earlier we used to say that like if this gene is there, this gene will make tall plant tall. But how it will make tall? Does the having a gene will do that? But because we know that what genes are, what these are present inside the chromosomes, and chromosomes are present in the cell. How each cell will have, how they know that which has to do what work? That is a major question now. And how they are actually making us to grow the height, right? This thing we're going to be learning and understanding in this molecular basis of inheritance, because these are molecules or a minute level things that are taking place inside our body or organism body. I hope this basic thing is understood to you. Starting with it. Now, molecules are what? Which molecules are working in it? So definitely the basic genes are present on what? It is present on a genetic material. The genetic materials could be of two types. Till now, if you have studied a little bit, you know that there are two different types of genes that are present. Sorry, two types of genetic material that is present. What are two types of genetic materials? Either it would be DNA or RNA. We know that the most abundant one is what? DNA, which is present in all the organisms except few viruses which are keeping RNA as their genetic material. What problem it creates, we will learn it. Why DNA is more compared to RNA? Let's learn. These questions which are coming us right now, that okay, this chapter has to do something with the DNA and how DNA is working to give us something, we will learn it here. I hope this basic intro is done. Now, if you will ask that, sir, why this is even important for our medical journey? Because beta, you would be studying about the biochemistry. Over there, these basic molecules are definitely gonna come. But at least you should know, like if you go into the PGs and all, you will do a lot of researches also. In that research, you should at least know now where this is working. I will let you know that what are the things that will help you out in your medical fields with this, but it's an important chapter for you. Okay, so molecular basis of inheritance, we understand that there is something DNA, something RNA thing. Okay, so first thing is what? That we are talking about genetic material. So let's just talk about that only, how that thing was actually being identified. Now what happened, there was a person, F. Misher. He identified this genetic material DNA present inside the cell in the nucleus. Okay, and he called it what? Nuclein. Okay, later on it was found out that it has what? Acidic nature. Why it has acidic natures? Because of presence of phosphate group into it. Okay, so later on it was termed as nucleic acid. Okay, so this is the basic thing. So let's just be starting with that only. Okay, so our chapter is molecular basis of inheritance. So what we have studied, that there is a person, F. Misher, first observed DNA and termed it what? Nuclein. Termed Nuclein. Later, Altman found out its acidic nature and termed it what? Nucleic acid. Okay, termed it as nucleic acid. Now the next thing is there are two types of nucleic acid, right? DNA and RNA. Okay, so nucleic acid is of two types. We have DNA and we have RNA. DNA is what? Deoxyribonucleic acid. RNA is ribonucleic acid. Okay, so now different species has both. Normally this is found in most of the organisms, or in fact all of the organisms. RNA in few of the viruses. Okay, let's just see that. Now what happened? They are made up of what? DNA and RNA. DNA and RNA are made up of nucleotides. Means what? Like if you see this diagram, over here what is it? This is the DNA we used to make like this. But what is this? This thing is what? This looks like an individual thing, right? This is one part, this is one part, one part, one part. And they are joined together like this. Can you see this? Can you appreciate this thing? This is what? This is the one part. This is the one unit, one unit, one unit. These multiple units comes together and form a long structure like this, which is helically coiled. Helically coiled means like this, like the braids which girls has. You might have seen that, no? They make the braids. Okay, so that is what, there are like hair strands and they are moving the hair strands like that. So it is like that. Okay, so this is the best way of understanding it. So girl's braid is what? How the DNA is helically structured like this whole thing. Okay, but individually they are made up of something. What is that? That is nucleotides. Okay, if you remember little bit from your 11th class biomolecules, you know that what is nucleotides. But if not, don't worry. Who is studying in 11th? No one is studying in 11th. So we're going to be studying it here itself. So how we're going to be studying it? We're going to be studying that how this nucleotides are being formed, what is their compositions, how they look like, means whole chemistry. In short, now the biology is contradicting with the chemistry. And with that, we come to know that, okay, chemistry and biology is a little bit linked. Okay, now you will like, don't start it again, but we have to do it better. Okay, but I'm making sure that this is easier for you. Okay, so before to go with it, so we know that these nucleic acids are made up of what? Nucleotides. Okay, so here we're going to be writing that out. Made up of many nucleotides join together. So we understand this thing. Little bit you are able to understand. Okay, so in the individual things, it will join together and form the structure. Let's try to form it. Okay, now what is our question is, we have to form it. Before form it, let's see that DNA and RNA, that this every organism has same number of nucleotides. The answer is no. There are different types of there are different number of nucleotides present in the organisms, and that is what defining thing. No two different species have the same looking nucleotides, or the means same type of a genetic material. Nucleotides are wrong, same type of genetic material. Okay, there will be a difference. Okay, like for example, our genetic material and a chimpanzee genetic material has 2% difference. But difference is there now. That's why it's making two different species. And that 2% difference also beta did what? Chimpanzees leads to the cages. We have captivated them in an area. But we are going beyond the earth, away from the earth, right? So this is what? This is just a 2% difference. Think about that. If some another species which has a DNA 2% or 1% more better than us, what will happen? Then we will be like chimpanzee to them. Okay, so this happens to it. I'm not terrifying you, but I'm telling you the facts. Okay, so we have to think in that way this chapter. Okay, not just learning from this, but the understanding the everything, because we need to make ourselves super good for our academic exams also, right? Conceptually sounds should be there. So let's just remember few of them. Okay, so for example, we have 174 bacteriophage. What is bacteriophage? Now, bacteriophage is what? It is the virus which is causing the disease in bacteria. That's why we call it bacteriophage. So here only we are writing it. So bacteriophage is what? Disease causing virus in bacteria. That is bacteriophage. Okay, this has how many nucleotides? It has almost 5, 3, 8, 6 bases. When I'm saying bases means what? Nucleotide bases. Okay, so why we call it bases? Because they are under the category of nitrogenous bases, means they have nitrogen present into it, and bases are basic units. Okay, so we call it what? Nitrogenous bases. Okay, so when the bases are written means they are calling what? Nitrogenous bases or the nucleotides. In other words, nucleotides. Okay, remember this point in your examination. Bases word could be changed with the nucleotides. Okay, clear with it. Now, when we are saying bases, it simply means that the base. Okay, let's just come back later on. Okay, coming up on the more examples, we have lambda phase. Okay, so in the lambda phase, we have 48502 base pairs. Okay, this is something different. We have bases, we have base pairs, which means what? It simply means that base pair means that it has a pairing, means it has one pair with the other one. Okay, means they are in the what? In a couple manner. Okay, means how many nucleotides would be there? The nucleotides would be double the number of base pair. So you understand that what is the base means and base pair? Base pair means they are in the form of what? A couple. Now couple means what? One person is here, other person is here. They join like this, another join like this, another join like this. Means multiple things are being joined. This is one base, this is another base. They're making pair. Okay, so means they are going for some kind of a ball dance, if you remember it like that, or if they are going for a couple dance. So couples will do what? They will find each other and they make a pair and they join up, right? Now the question is that the nucleotides would be how much? The nucleotides would be double, because base pair means one base pair has two nucleotides. So one base pair is equals to 2 nucleotides. So how many nucleotides would it has? It will be having that 4850 into 2 nucleotides will be there. So this can also be asked to you. So remember these words. Like now you will like, sir, how we will remember these words? Beta, this you have to remember it. There is no mnemonic to learn it. So you will remember these number of bases, and these number of bases, if you remember it, these are given in NCERT. So match the following can come. So even this in this form it can come. So we are learning with the understanding. So understood what is the difference between base and base pair. Base pair means two nucleotides are joined together and one. One more thing, if bases are there means they are singly present, means 1, 2, 3, 4, means it's a single strand which is present. So means in the 174 bacteriophage, it has what? Single strand of genetic material present, whereas in the lambda phase it has a double strand present. So one more thing with this base and base pair, we understand what? That this is what? Single strand, and this is what? Double strand. What it is? DNA only. Okay, so this you have to remember that all of them are what? Having the DNA as a genetic material. Clear? Then after lambda phase we have E. coli, Escherichia coli, which is present inside our gut also and causes some diseases also. They have 4.6 into 10 raised to power 6 base pair. Base pair means double stranded, and this has DNA. Because E. coli is what? Bacteria. Bacteria will have a DNA only. Bacteria means kingdom Monera, Protista, fungi, Animalia, Plantae, they have DNA only. Just the questions would be, there is a tougher to you, that is in the viruses. Some viruses has what? RNA present. Okay, next we have one more example, that is human genome. Human genome. What is the meaning of word genome? Genome means haploid. Haploid are what? In our body, in my body, in your body, where are haploid cells present? We said now we are diploid. I told you this thing in the last genetics chapter. That is, what is haploid cells in our body? Only our gametes. So means we are talking about the gamete over here. Okay, so genome means what? Haploid. Plus this means what? Gametes. So gametes has how many of the base pair? 3.3 into 10 raised to power 9 base pairs. This is also double-stranded DNA. Okay, now the next thing is that if this is the genome, then what is our diploid cell content is? It's doubled. Okay, so means over here, if someone by chance asked you, then what is the diploid or somatic cells? You remember these words, na? We have already discussed these words in the genetics chapter. Okay, here again I'm telling you means your other cell, like this cell, all the other cell except in me sperm cells, in females ovas. Okay, they have what? They are somatic cells. We call them somatic cells, vegetative cells. Vegetative word is even used for more for the plants, not for the humans or animals. For animals we use the somatic words. Somatic are what? These are not the haploid cells. These are diploid cells. So how you will remember it? You will 2 into 3.3 into 10 raised to power 9 base pairs. That is 6.6. Understood? Remember it. The concepts has been told to you. The things has been marked to you. Okay, you can easily do it. Okay, just remember the number of things. Okay. With this, let's just go into that. Okay, there are a lot of DNA is coming up. Let's just study about DNA and let's see that how the RNA is present. Now, remember little things about it. The DNA is made up of what? Some basic structures. So over here we said na, it is joined by many number of nucleotides are joined with it, right? Let's just make those nucleotides. So everything starts from what? From the nucleotides. Okay, so we're gonna be starting with the topic that is structure of DNA. Structure of DNA. Now, in DNA, one thing is for sure, DNA is never single strand. It is made up of 2 strands. Okay, so it is what? It is double-stranded. Means made up of 2 polynucleotide chain. When I'm saying 2 poly, means 2 chains are there. Polynucleotides. Poly means many nucleotides, and different types of nucleotides are working over it. What are they? We will learn it. Don't worry. Okay. So we understand that it has 2 strands. Okay, and they are made up of multiple nucleotides which are joined together. Able to imagine a little? Okay. So imagination should be very high in this chapter. Okay, we're gonna be building that imagination. Don't worry if your focus, if your imagination power has been taken away by AI, we make sure it comes back into the classroom over here. Okay, so now the next thing is that, so 2 polynucleotide chains, and what the chains consist of. It consists of, first, okay, better would be like this. So nucleotide consists of 3 things. Okay, so we're gonna be writing it like this. Nucleotide, the basic unit. This is made up of 3 things. What are the 3 things? There is presence of a phosphate, there has to be presence of a sugar. Since it is a DNA, it's gonna be deoxyribonucleic ribose sugar, and there should be presence of a nitrogenous base, means it has a nitrogen present into it. Okay, that's why nitrogen is so important for our diet, because if the nitrogen won't be there, that DNA won't be forming in our body. DNA formed by pre-existing DNA, that we will learn. Okay, now let's just see that, okay, there are three things: nitrogenous base nitrogenous base is required, sugar is required, and phosphate is required. Initially what happened, this nitrogenous base joins with the sugar, and this phenomena and this structure that is formed, this is when the phosphate is not joined with, we call it nucleoside. Okay, so what I said beta, that is, okay, not this one, that these first these two will join, and once these two will join, we call them what? We call them nucleoside. So if someone will ask, what is nucleoside? Nucleoside is sugar joined with what? Nitrogenous base. That is nucleoside. With this nucleoside, if the phosphate is also joined, we call it nucleotide. How you will remember it? See, there is a trick for it. If you see that nucleotide, T, nucleoside, S, this T means what? Third. S means what? Second. Second means what? 2. Third means what? 3. So in nucleotide, 3 things will come. In nucleoside, 2 things will come. What is not present? Phosphate is not present. In what? In one, in other it's not. Easier to remember. Nucleotide and nucleoside, its naming also we will learn it, don't worry. Okay, so this is one thing. Now we said that in the DNA nitrogenous bases are there, but nitrogenous bases is also further divided. Let's just study about them. The nitrogenous bases are basically divided into 2 categories. This is very important because in biochemistry this will come. Like when you will go in the first year of your MBBS, biochemistry is the subject where you should know about these things. DNA of course, and nitrogenous for sure, because from this nitrogenous base only your urine formation is also there. Okay, and you will be like that, okay sir, if it is like that, then this is so much important. How do you know? Beta, I went through that college of dentistry. Over there the subject was biochemistry for us also, and my biochemistry was horrible. Okay, it was so horrible. Later on I have learned all those things when I was teaching you. Okay, but at that time I was so horrible in it. Okay, my friends still, I remember that how me and my friend was totally blank in the examination. Okay, not just the written examination, but in the viva, we don't know anything, that how this nitrogenous bases are doing their work. But that's a story for some other day. Even if you want, I can help you out with the biochemistry also later on. But let's just come back. So nitrogenous bases are 2 types, purines and pyrimidines. Okay, so nitrogenous bases are what? Purines and pyrimidines. Now these are what? These are rings. Now the purine is heterocyclic ring, means it is made up of 2 different rings. Hetero means, and both the rings are different. One ring is 6-membered, other ring is 5-membered. Whereas pyrimidine is what? It is one single ring member, 6 components are present on each corner, and it is what? It looks like that. In the purine we have substituted the 5-member ring. Understood? Okay, this thing is there. So purines and pyrimidines. In purines we have what? We have 2 options. What are they? We have adenine and guanine. Where pyrimidine, in DNA we have 2 options here also. Actually there are 3 pyrimidines, but in DNA only 2 are present, not the third one. Okay, so what are that? There is what? We have cytosine and thymine. Now if someone will ask, like in DNA, which pyrimidine is not present? Uracil. Okay, so over here what? Uracil absent. Understood? In DNA, pyrimidine. Now how they looks like if they had to make a bond, let's just try to make the bonds of them by knowing that individually how they look like. So phosphate is what? Simplest thing. So phosphate looks like what? It is what? P, sorry, if I have to make it at the this way. So phosphate is P O O O and O. Like this way it is there. Okay. So normally it is 2 negative. This is how it will look like. How a sugar will look like? Let's see. Sugar. The sugar I am making it over here. So sugar will be like this. Pentose sugar means 5 carbon. Pentose word penta. Okay, if you know a little bit of organic chemistry, you can work it out. Okay, so this is what 1 number carbon, 2 number carbon, 3 number carbon, 4 number carbon, and 5th carbon is like this. Okay, now these each carbon will have what? Valencies. Okay, so like this, and this is what? CH2OH. This is OH, H, OH, H, H. Here what happened at the second position, there is what? H present. Here what happened, beta? If we are comparing it with the ribose sugar, like the name itself saying that deoxy. Deoxy means removal of OH group. So basically, in reality, what happened? This ribose sugar has OH over here present. But to make it deoxyribose, it goes away. Now someone will ask like, why it is going away? It is going away to make this sugar more stable. We will learn those things. Okay. So for now, we remember it like this. This is the ribose sugar. Now let's see about the purines and pyrimidines. First about the purines. Now I told you that it is what? Heterocyclic ring, whereas pyrimidine is what? Single ring. Okay. So we're going to be starting with the purines. Okay. So we have purines. Purines are what? It is heterocyclic or double ring. Okay, now you have to remember that at nitrogenous bases where they are present. They are present at 1, 3, 7th and 9th position. It is common having nitrogen at position number. Like remember now, I write their position also 1, 2, 3, 4, 5. So here also having nitrogen at position 1, 3, 7 and 9. Let's just make it. 2 rings are there. Okay, so the rings would be like we're going to be making first ring, then we're going to be adding the things. Okay, so see over here. First I am making adenine. Adenine over here. So first ring. Okay, and the second ring. If you see the first ring which I have made, it has what? It has a 6 members, whereas the next has the 5 members. 2 members are being shared by both the rings. Okay, so we're gonna be saying it like that. Now we have to write the numbering of it. So the numbering will start something like this: 1, 2, 3, 4, 5, 6. First the numbering in 1 will be completed, then the numbering in second will be started. The numbering will start from here: 7, 8 and 9. Next, what we know? We know that there will be nitrogen present at 1, 3, 7 and 9 position. So we're going to be writing our nitrogen now. Okay. So 1, 3, 7 and 9th position. This is what where our nitrogen is present. Now the next thing, at the other places carbon is present. Okay, understood. Okay. Next thing is that we will be seeing that in this ring where there will be double bonds present. So the next thing is double bonds. So if you see that first double bond would be here. Okay. The next double bond would be this. Next would be this. And the last would be this. So 4 double bonds would be there. With double bonds, its valency would be over. Okay, everywhere where nothing is written means H is there, right? So valency of carbon, nitrogen, everything is being fulfilled over here. You can check it yourselves. Moving on to the next thing, that is the extra groups that is present to it, functional groups. So at the 6th position over here, there is an amino group present. So it is written by like this, NH2. Okay, this is your adenine. Okay, after adenine, the next is what? Next is our guanine. So we're gonna be making the guanine next. So guanine. So again, everything will start from what? The basic thing is what? Heterocyclic ring, nitrogen at the required positions, right? So we're gonna be writing it that. Okay, so what we have? We have a, okay, this don't look right. We have first ring, this is our second ring. Okay, now nitrogen, like numbering, we're gonna be doing the same numbering we have to do. 1, 1, 2, 3, 4, 5, 6, then 7, 8, and 9. Then we know that nitrogen would be present. So where is the nitrogen present? We are writing it with this. So nitrogen is present at the 1 position, 3rd position, 7th position, and the 9th position. Okay, now after this, we're gonna be seeing that where would be the double bonds present in this case. So here there is a little change. Okay, so first double bond is present over here, next here, and here. Okay, here, and what is this? This and this. So means what is the difference? That at 1 and 6 position there is no double bond. Means here we have to give a certain type of what we say that at the 6th position probably we have to give a certain type of a functional group which complete the valencies. So how much valency at the 6th position carbon will have? There's a 2 valency that has to be completed. So we give a something which has a double bond to it, and that is oxygen. Okay, along with that, at the 2nd position also we will substitute the H over here from a to a amino group. That's it. This is your guanine. Okay, I hope this thing is understood to you. Coming up on the pyrimidines. Okay, so pyrimidines over here, pyrimidine. Okay, this I'm not on the camera when I'm writing. Okay, perfect. Pyrimidine. Now the pyrimidine is what? Single ring. Single ring is present with nitrogen at 1, 3 position. So means because second ring is not there, na? Because we know that in the second ring the nitrogen would be at 7th and 9th position, but over here it would be at 1st and the 3rd position only. Okay, please let me know that whether you understand my English or not. Okay, if not, then let me know that I will change or slow down the speed. Okay, 12th class students. So we have what? Number 1, we have cytosine. Okay, in cytosine single ring is there, so we will make a single ring. Okay, this don't look good. Okay, now numbering, we're gonna be putting up the numbering. Same number would be there. Okay, 1, 2, 3, 4, 5, and 6. Okay, now we know that where the nitrogen is present at the 1 and 3 position. So 1 position and the 3rd position. Here we're gonna be giving our nitrogen. Moving on, what would be the double bonds where that would be present? Okay, so your double bonds would be present at this location only, at this 5 and 4 position, and then you will have to give the groups. If we see at the first, then at the position of 2 there is 2 valency left. So how we will complete it? We will be adding what? We would be adding the O to it. Okay, then at the sixth position also 2 valencies left. So we're going to be adding what? We're going to be adding this. Now 1, 2, 6 is completed. In 3, 2 bonds is there, 1 is left out, that's going to be H. At fourth position, 3 bonds are there with the carbon, 1 bond would be the H. And at this fifth carbon, there is only 3 bonds present. The last one will be H. No, it would be methyl group, CH3. Okay, this is cytosine. Okay, next come our thymine. Sorry, this is thymine. Okay, I made it wrong. This is thymine. Okay, thymine. Then we're going to be making our what? Cytosine. Cytosine. Same thing, we're going to be making this ring. Okay, now on this we remember this would be the numbering. Then we have to remember there where would be the nitrogen. Nitrogen would be here. Then we're going to be seeing where would be the double bonds. Double bonds would be here and here. Okay, we know that everywhere in all of these fourth and fifth carbon has double bonds present. Okay, next we're going to be adding up completing the valency. So first valency over here. So 1, 2 and 3 completed. Fourth also completed. Fifth also completed. Sixth is the place where ammonia will be present. Okay, this is what you have to remember. This can come in your syllabus, so make it again and again, then only you will be able to recall. I have give you a way, step by step way how you will remember it. So how you will remember it? First you will remember that which 2 rings or single ring. Then you will remember the numbering. After numbering you will put where the nitrogen would be there. After putting the nitrogen you will remember. Now there comes the remembering factor. First, where would be the double bonds? You know that there will be 4, 5, definitely there will be a double bond in all of them. Other you have to remember. After that you have to see that where you have to give the functional group. 1 functional group, 2 functional group, and how many it is. Okay, so that's it about the purines and pyrimidine structures. Now we're going to be seeing that how the DNA looks like. Now let's just try to make a structure, means a nucleotide or a nucleoside. So when I'm saying nucleoside, it means that of course, because first what, when a nitrogenous base is joined with the sugar, it forms what? Nucleoside. So let's say the nucleoside, and then comparatively we will even make what? A nucleotide also. And whatever we will make, we have to give the name also. So we will try to give that also. Let's just see into it. So there will be what? There is sugar in the bit. So we're going to be making a sugar. So this is how the sugar look like. Okay, 1 number carbon, 2nd number carbon, 3rd number carbon, 4 number carbon, and this is the 5th carbon. I'm gonna make it opened up. Okay, COH HH. This is H OH H OH and H. Okay, this is your what? This is your sugar. Now with the sugar, what would be added? I told you that if we have to make a nucleoside, we have to add what? A nitrogenous base. So let's take any example of a nitrogenous base. Okay, so this would be joined with this. So over here, if I'm making a nitrogenous base, it will join. So we have what? We have either purine or we have pyrimidine. Now the purine, which part of the purine will make bond? There will be, in the purine case, there will be N9, means 9th position nitrogen will be the part of the bond over here. And if it is a pyrimidine, then it is N1. The first position nitrogen would be responsible for what? For the bond formation. So in pyrimidine, cytosine or thymine will have the first number nitrogen bond formation. And in the purine, there will be what? There will be only 9th number. So over here, I am just simply writing it what happened basically. This thing will go and the H bounded with the nitrogen goes, we will make it, don't worry, goes and form this structure. So if I'm making any nitrogenous base, let's take it this. Okay, this is what one pyrimidine, normal examples. This is what first number nitrogen. So this first number nitrogen will have what? Its own hydrogen. So what basically gonna happen? This thing, both will join and leaves like a water. Okay, this color is already here. So what happened? These both will leave. So water will go away and this will leads to what? There is a one valency, okay, positive valency here and a negative valency here. It will do what? It will better go and join with it. So this bond formation will take place. Now this bond name that you have to remember what it is, since it has what? A sugar involved. So for sugar we use glyco what? Plus nitrogenous base involved means N. Okay, so the name is N glycosidic bond. Did you understand what is happening over here? What I said that in the first carbon of the sugar, it has OH group and the H group, whereas attached to it, okay, functional groups. Then with the nitrogenous in the nitrogenous base, the nitrogen at first position in case of pyrimidine or ninth position in case of purines, they have what? With nitrogen, hydrogen is present. This OH of the sugar, whereas H of the nitrogenous base, they make a bond and release like a dehydration reaction. When they will release, they create what? Valency over here. Okay, this valency will be completed by making a bond over here. This kind of a bond is called as what? N-glycosidic bond. Okay, N is for the nitrogenous base, glyco word is for the sugar. Okay, now this thing that has been formed, it is what called as, this thing is called as nucleotide. Nucleoside. So nucleoside has what? Sugar plus nitrogenous base. So it has what? Sugar plus nitrogenous base. Now how do we name it? We simply name it as a cell. Okay, means what? For example, if we have adenine, its nucleoside would be what? Adenosine. Or if you want to write it, deoxyadenosine. Adenosine, you can't tell it whether it's the ribo or this. Normally we take it, this is what? It is the ribonucleic acid, has ribo sugar. Okay, so deoxyribo sugar means which has a this deoxy sugar na, this is deoxy sugar at this position, right? So this thing will be there. Now if it is if it's guanine, then what will we say that? We will say that guanosine, or we will call it deoxyguanosine. If we have what? We have cytosine, then we will call it cytidine. Not sine, but dene, cytidine, or we call it deoxycytidine. Okay, next is we have thymine. Thymine will also make this bond, so we call them thymidine, or it is called as deoxythymidine. I hope this thing is understood. Now what happened next thing? The next thing is that there will be joining of phosphate. Now where the phosphate will join? Phosphate will join at the 5th carbon of the sugar. Okay, so it will happen what? It will join over here. So sugar will be joining over here. How it will join, beta? Let's see. Now, what happened? That this bond will be broken up. Okay, one bond will be broken up. Better. Like this. It will be broken up. And it will what? It will dehydration reactions in the body. Most of the reactions are dehydration reactions to make the bond. Okay, just remember it. Okay, so what will happen? This will create what? A valency over here. This will create a valency over here. So this will be what? Positive. This would be a negative. So it will do what? It will make a bond. This bond name is what? Here the phospho is involved. This whole thing is what, beta? This is phosphate. Okay, this is what? This is O is coming, and then a sugar component is coming. If there is a carbon in which, like, carbon is there, other thing is there, and in between oxygen comes in the bonding. This is what? Esters, if you remember from your 10th class. So this is the esters, organic chemistry. So we call it phospho. They are what? Phosphoester bond. Because POC bond is seen over here. Okay, so ester is coming over here. Understood? So if this thing is coming up, we will say what? Now we will say that this whole thing is what? Okay. This here, all the three things are what? Nucleotide. Means it consists of sugar plus nitrogenous base plus phosphate. And if someone will say that, what would be their naming? Because naming is super important. So the naming would be the like adenosine or deoxyadenosine will always be there, but the phosphate group is added to it. Okay, single phosphate is added to it. So we'll call it monophosphate words to it. So what it is deoxyadenosine monophosphate. The phosphate is being added, so we will add that word. Same for the guanosine, that is deoxyguanosine monophosphate. Understood? The name is very easy. If you understand the nomenclature now in chemistry, it's very easy. If it's tough for you, then let me know that I will help you with the short. Okay, then comes the deoxycytidine monophosphate, or it is deoxythymidine monophosphate. Now over here, since I told you what, your DNA is what? Acidic. Why it is acidic? Because of the presence of this structure. This phosphate group has what? Acidic behavior. Phosphatic acid or phosphoric acid, you know na? So that phosphoric acid is giving it what? An acidic environment. So a acid suffix name will also be given to it, to this nucleotides. The nucleotide has acid substitute name, not the nucleoside, because acid has what? Phosphate. Okay, so what we will call it? So we will call it deoxyadenilic acid. Remember now, we used to call it carboxylic acid. Okay, so ilic acid word is coming up. So here also it is coming up. Deoxy, okay, acid, deoxyguanilic guanilic acid. Same goes with cytidilic acid, thymidilic acid. Okay, so this thing will be there. So this is what? Cytidilic acid, deoxythymidilic acid. Okay, so these namings you have to remember because they can directly come up that they will give you the names. If you understand the naming and the nomenclature, then you can easily match or find the answers to it. So it is important for you to understand from the question point of view. Okay, so now the next thing is which you should remember is that okay, these are joined together, but how they are joined together, okay, and what are their structure finally look like. Of course, in the front pictures, when I give it to you, when we started this chapter, there was a picture of a DNA look like this, helical and all. But how do we know that? We know that with the help of two persons who give a proposed theory. But the theory was made on the experiment by Wilkins and the Rosalind. Okay, Roselkind's. Okay, so they did what? They did certain experiment on what X-ray diffraction of the DNA of the genetic material, and they found out that how their composition it is, and then they made a thing. Normally it is said that they actually did the experiment, but Watson and Crick took away the information, okay, and or they stole the information and then presented it to the world. Okay, but they also acknowledges that this is based on the information or the experiments by those two scientists. Okay, so if someone will ask like Watson and Crick model is based on what? It is based on of X-ray diffraction experiment. Okay, so those two things are there for you to remember it. Okay, next is okay, so let's just see that how it looks like. Before to go into it, okay, I want to tell you that how these chains are, like we have made what? We have made this structure that one nucleotide is like this. So one nucleotide, next nucleotide, next nucleotide, next nucleotide. That's how two different strings gonna be made, right? So let's just see that how that strings are being made. Okay, one by one. Okay, so that is what it is, what Watson and Crick experiment. Okay, so we're gonna be writing about that. So that is our topic, Watson and Crick model of DNA. In Watson and Crick model of DNA, so we have to know a lot of things about it. Okay, so let's just one by one know it, and then we will go into deeper part of it. Okay, so Watson and Crick model based on the observations from X-ray diffraction experiment by Wilkins and Rosalind. Okay, now in this experiment they're able to find things. Okay, now the DNA is of different types. Okay, so which DNA they have done experiment on? That is a beta DNA. Okay, what it is, its features we're gonna be seeing up. Okay, later on what are the other types of DNA, their features we will also study simultaneously. Okay, so in this experiment what is happening, let's see. So how it looks like. Okay, so we need one nucleotides, we have to keep on joining the nucleotides. We know that nucleotides are basic unit, right? Now in this nucleotides, how they are arranged to it. So let's just make it. So we have what? We have a, this is the first chain I am making and the second chain I am making. It is a double chain structure. Okay, so that we're gonna be making. Okay, with this lot of things we're gonna be understanding. This is the one chain. Okay, and in the one chain I'm gonna make, then I'm gonna make the second chain. Okay, so the first chain is like, we're gonna see now how the one chain is being made. Okay, so this is what, this is a sugar. This is 1 number carbon, 2 number carbon, 3 number carbon, 4 number carbon, and this is gonna be the 5th number carbon, right? Same 1 number carbon, 2 number carbon, 3 number carbon, 4 number carbon, and this is the 5th number carbon. Now this thing, this first number carbon will be joined with what? A nitrogenous group. Okay, so I'm gonna be making a nitrogenous group over here. Okay, so see over here, I'm making 1, 1, 2, 3, 4, 5, 6, 6. Okay, and here I'm making another carbon. That's it. Okay, so this is another. So if you see the first diagram, it is what? It is what? A single ring, means it is a pyrimidine. The next one is purine. Of course, you can just tell this purine and pyrimidine only because the further functional groups are not added. Once I will add the functional group, you are able to identify that whether it is what type of purine or what type of pyrimidine. Okay, so let's just see that I am adding the things. So over here, I added what? I added a double bond here. Okay, not here, here. Yeah, here. Here I'm gonna be adding an O, and here also O. Okay, so this is what? This is a thymine. Okay, so this is thymine. Now this structure which I'm gonna be making up now, this is this. I make O, I make this, and NH2. This is what? This is guanine. Okay, so this is guanine. Now we have the phosphate also attached to it, so I'm gonna making a phosphate over here. So phosphate would be joined like this: O, P, O, O, negative, negative, then O, P, O, O negative, O, like this. So these are what? 2 nitrogenous base. I told you what? 1 nitrogenous base, next, next, next. So they are what? They're building up once and again. Means when they are building up, they must be joined. How they will be joined? Let's just see that. So this is the first nitrogenous base. This is the second nitrogenous base. So what's gonna happen? The first nitrogenous base will join with the second nitrogenous base, which is present just next to it, with the help of what? If you see over here, this thing and this thing is closer. Means what gonna be happening over there? If you know the structure of it, you know that there is what? OH present and there is H present. Means if what I do, I basically make a bond over here. How I'm gonna making a bond over here? I'm gonna be making a bond over here with the help of what? Normally, you know what? That there is, if we see the normal structure, like OH is present over here, right? So we have to make it like negative also I'm showing, or OH also I'm showing. It means saying that hydrogen has been left from there. Okay, so what happened? See over here. Now what will happen? This 2 things will go. So here is what? Water removal. Okay, so if I have to make it, what I will do? I will remove this thing. So it removed. Okay, water removed. So what is left with us? We are left with this thing, that this phosphate which has a positive charge and this has a negative charge, which will make a bond over here. So ultimately what happened here? A bond formed by the removal of the water, and this is how one nucleotide joins with other, and the long chains will keep on forming. Now if you see what it looks like, if I have to just remove this, this thing is removed. So means POC bond is there. POC bond is on this side. So POC is this bond. POC is this bond. Means here what happened? Two phosphodiester bonds are there. So when the ester bonds are 2 in number, we use the word diester. So this bond which you are seeing over here, that has been formed. This bond name is what? This bond name is phosphodiester bond. Understood? So we have what? Phosphodiester bond that is being formed. Till now, how many types of bond we have studied? We studied some number 1, N-glycosidic bond. Where it is present? Here it is present. We have studied, and here also of course. Then the next bond that we studied is phosphoester bond. So this is phosphoester bond, phosphoester bond. This is also phosphoester bond. Then we have studied what? Phosphodiester, because here phosphate is making 2 ester bonds. So phosphodiester means 2 esters are present now, so diester. Then you're like, sir, why don't we add diphospho? Means 2 phospho are not there. Phospho is 1. Ester bond are 2. That's why we are saying that phosphodiester bond. Understood these bonds? They can even mark you and ask you that A, B, C, D. This is, they will mark it that A. This is, they mark it as B, and this is, they mark it as C. And they will say that which is which bond. For your board exams also, for your NEET also, this is very important to understand. Okay, we understand. Now what happened? We said that this looks like this. So there might be the initial point, like always when we have a like a line. We always say a line has an initial point and an end point, right? We used to write that this is a line, for example. You said that A and B. So A and B is what? It's starting and the ending point, or the 2 ends of it. This chain which you have just made, this gonna be what? A long chain over here. Means this is there, this is there, next would be there. Means long chain is there. This chain also should have an initiating and an ending points, or maybe the 2 ends. We have to name it A, B, C, D something, right? Here what we do, we name it 5' and 3'. How it is coming? See over here, beta, what is happening? If you see that this is what? 5th number carbon, which is making with the bond with the phosphate. Here, if you see that this is the 5th number carbon, which has a phosphate, which is not free, it is making a bond with the 3rd number carbon. Means it is involving this 5th number thing, 5th number this thing is involved somehow further, extending it. This is on the basis of sugar only, the naming which I am giving, on the basis of sugar. If the 5th carbon has phosphate which is free and can make a bond, we call it that 5' end. This is a 5' end. Okay, I'm going to be writing it with the white. This is what? 5' end. Whereas if you see on the other end, over here, this is what? Next nucleotide is absent. Means this 3 is still free, and if you put the next nucleotide, it will make a bond, right? So this free end on the other terminal is termed as 3'. This is how we have named this one strand as 5' to 3'. Will you remember it? Okay, now what happened? Now I said that DNA is double strand. So means what? This is the one strand. Another similar strand will be over here, but I have not made it. Let's try to make it. But here something is there. We said that is it anti-parallel. Anti-parallel means what? If this line is 5' to 3', the other line would be 3' to 5', opposite of it. Opposite means that we have to change the polarity now. So we will make it little different. So how are we going to be making it? Let's just see it. So what I do, I keep my pad like this now. Okay, so okay, my pad is also being done. They also done this like this. Okay, so it's like this. So now I'm going to be making a 5' over here and 3' over here. Before I am making anything, I have to remember one fact. What is the fact? The fact is that these nucleotides bond very specifically. When I'm saying that they are bonding very specifically means what? That adenine will always make bond with thymine, and cytosine always make bond with guanosine. Okay, means these nucleotides are very specific with whom they will make the bond. That will also confirm certain more things which we have to remember about this chapter, because this chapter is all about understanding the statements. If you understand my lectures, then you will understand the statements super well also. let's just see them. Okay, so we have what? We have to make a opposite of it. So we have thymine. Thymine's opposite would be what? Adenine, right? So we're gonna be making a adenine over here. So let's make the sugar. Since it is a reverse polarity thing, so we will gonna make it upside down. Okay, so this is 1, 2, 3, 4, and this is a 5th carbon. Okay, same over here we're gonna be making. First of all, they are equal size. Since this is, I'm making the drawing, I can't make it of equal size. That's why they look little bigger than usual. Okay, I'm just gonna making so that it is visible things. Okay, this is 1, 2, 3, 4, and the 5th carbon. This is 5 dash. Now we're gonna be making what? Its counterparts. Okay, so this is what this has a like adenine. Sorry, thymine has what? Adenine. So we're gonna be making a adenine. So adenine is what? It is a purine, means heterocyclic ring. 9th number carbon will be joining to it. Okay, so it will be something like this. Okay, and this is what? Guanine. Guanine will be mixed with the cytosine, which is what? A pyrimidine. Okay, so it will have what? N1. So this is 1, this is 2, this is 3. Nitrogen over here, this oxygen comes over here, this bond NH3 is here, and they have a double bond like this. Yeah, this is how a cytosine will look like. This is, I'm gonna making a adenine, so cut like this, and one would be, wait, I made something wrong. This would be N here, and this will join like this. This will be the bonded. Then we have N over here, N over here, and a bond like this. Then this is NH2. Adenine is easy. Okay, adenine. Okay, you will make again and again, you will able to do it. Okay, let's just go into it. So it is there. Now I told you what? Wherever nothing is written means what? There is a hydrogen. Okay, so of course this other branch, of course I didn't made it over here, but this look over here, the phosphate would be there. So this is what? Phosphate. What is this? This look, phosphate. We'll be making a bond with it. Okay, so I have just made it a simply, not making a elaborated form. Okay, so phosphate is here, and next the phosphate is not there. Okay, so let's just not dive into the deeper section of it. Let's just see that they are bonding to it. So two anti-parallel chains I have made it. Now how these two chains are joined together, that they are staying together. We said that it's staying together, joined to it. They are joined with the help of hydrogen bonds. Means what? Hydrogen bonds, if you remember what it is, that nitrogen, oxygen, and fluorine, they have a higher electronegativity, which attracts the bonds of the hydrogens. Okay, electrons of the hydrogens, and it makes what? A hydrogen bond, right? Here also that happens. So now I'm giving you something which you have to remember. Now when I told that adenine will make bond with thymine, cytosine will make bond with guanine. Now adenine and thymine will make 2 hydrogen bonds, whereas cytosine and guanine will make 3 hydrogen bonds among themselves. And now understand that so much of hydrogen, and hydrogen bond always forms when there is a close affinity between these molecules, nitrogen, oxygen, fluorine with the hydrogen of the opposite over there. You know this thing, right? So let's just see that what gonna happen over here. So which color? Now let's use this. Okay, all the colors has been used up. What do I use? So we're gonna be using this very bright green. Okay, so now let's just see. Hydrogen is there. You know that wherever not written, that means hydrogen to complete the valencies. So adenine and thymine, this is what adenine and thymine thing. They will have 2 hydrogen bonds. Okay, let's just see that what gonna be there. First bond will be between what? Okay. Okay, let me just make it a little better. Okay, little. Okay, let's just where is the yellow color? Okay, okay. Wait a second, I'm just correcting the diagram a little bit, changing the position. Okay, so that's easier for us. Now it's easier for us. Let's just do it. So it will make two bonds. What would be the two bonds? One bond would be between this oxygen and this hydrogen of nitrogen. Okay, so if I have to, okay, I should open it up. This is NH. Okay, so let's just see this. So what's gonna happen, beta? This will be going and joining with this. Let's just use this thing. Okay, so this gonna be making a bond with this. So this is one hydrogen bond. Another hydrogen bond will be the nitrogen of this and the other. Okay, so over here, this nitrogen will have what? It will have a hydrogen with it, right? So what it will do? It will go and join with this. Means what? This purple color thing which I have made, that is what? That is the hydrogen bond. So this is what? H bond. H bond. Same thing, now we have to make the three bonds between it. Just look at that. No one gonna ask you this thing. Okay, if you just directly made it also, now that also believable. Okay, here I'm just showing you what is happening, where it is happening. Same thing in this over here. If you see the guanosine and the cytosine, so there will be three bonds. So one bond is being made between this. One bond is this. Okay, another bond is this, and one bond is this. These three bonds, okay, that is being made. Okay, I just didn't make the diagram that good way. So if you made in the diagram in the good way, it will come up. Okay, so this is also what, beta? This is also the hydrogen bonds. So here we have what? Three hydrogen bonds present. This you have to remember, two hydrogen bond, three hydrogen bonds. Understood? This is how a long chain will be formed. Okay, that we have to remember. Till now we have seen a lot of things. Let's just summarize that what is the final version of this thing and see that how the DNA model will finally look like. This we have seen the basic version, what are the bonds are present. So we studied a lot of bonds. Okay, so you have to remember that all the bonds which we have studied. Moving on, that is what the features of it. Okay, so we know that there is what? There is adenine, thymine, guanine, all of those things, right? Let's just write about it. Okay, that what is their bonds and everything which is possible, and then we will go with what our next thing, that is Char-gaff's rule, which we will come up. Okay, so we're gonna be doing that. We knows that certain points which we're gonna be writing up. So I'm just keeping it like this. Okay, so certain points that we're gonna be writing it up. What are those? DNA is double helical structure. Okay, where two helix runs anti-parallel to it. Where two helix runs, or two strands run anti-parallel to each other. 5' to 3' if you remember. To each other. Okay, so if I have to make it like this, a basic thing, I will be writing it 5' to 3' and opposite vice versa. Okay, and these two helicals are coiled. Okay, these helix are spirally coiled spirally coiled around an imaginary axis. Okay, so we need a coiling of it. So I'm not gonna making it. I'm gonna be taking a video of. I'm gonna be taking a photo of it. Okay, so that it's easier for us to complete and compile. Okay, this is fine, right? So okay, not a good photo though. We're gonna be checking another one. So if you see this is what two helical structures which are coiled like this, right? So this is what, if we have to make marks up certain things over it, so we can simply mark it. This is, if this is the 5' end beta, this is 5' end, its opposite end over here, this is 3', and this is vice versa. Okay, so we wrote the two things and they are spiral around an axis. Okay, next pointers that we're gonna be remembering about it, that is what? That is that the linkage between the two nucleotides consists of a phosphate linkage. Okay, so means this nucleotide, it is joined by what? Phosphate group, and we call it what? Phosphodiester. Okay, so these links, I'm going to be writing it up. Okay, the linkage we have already discussed, we are just writing it up and summing it up. Okay, the linkage between two nucleotides consist of a phosphodiester bond. You know that what it is, how it is came, how we name it, right? Is formed between C3 and C5 of different deoxyribose sugar of two adjacent nucleotide. Okay, are you able to understand? I'm explaining it again. You know that linkage is what? This C3 and C5 is making the linkage. This is what they are saying it, adjacent. Okay, I hope this thing is understood to you. Moving on to the next thing, that is one end of the polynucleotides has a sugar residue at 3', so we call it 3' polarity. Other is 5' polarity. Okay, so one end of polynucleotide chain has a sugar residue with third carbon not linked to another another nucleotide having free 3 OH. At the 3 OH functional group is there, na? 3 OH group, and other end has sugar residue with fifth position carbon linked to phosphate group, thus showing polarity as 3' and 5' respectively. Very easy, we have discussed it. I have just explained it in the written form also. Okay, now the next thing is the two polynucleotide chains, are joined together by hydrogen bonds. We have the hydrogen bonds. Okay. Adenine has thymine, guanine has cytosine. We have even written as two hydrogen bonds present between them, and there is three hydrogen bonds present between them. This also confirms one more thing which Char-gaffs give, which we will learn later. That is what, that if you see that adenine is what? It is a purine. Thymine is what? It is a pyrimidine. Cytosine is what? It is a pyrimidine. Guanine is what? It's a purine. Means the bonding will always take place between a pyrimidine and purine, not same purine or pyrimidine. And bonding always takes place between purine with pyrimidine only. Purine with pyrimidine only. Okay, I'm gonna be making a cut section so that no one get confused. This thing is there. Okay, we understand this thing. Then then we're going to be learning about the structure of it, that how our DNA look like. This we have already seen this thing, but we will go further into it. But before that, we would be finishing it up with the Char-gaffs rule. Okay, so these things, like all the things Watson and Crick is what? They are taking the things from the other people's. So one another person was there who gave what? Char-gaffs rule. Okay, so what is Char-gaffs rules? Char-gaffs rule is a phenomena. He said that purine will always make with the pyrimidines. So means in a DNA, the number of purines will be equal to number of pyrimidine. This is a very important question. So we're going to be solving certain questions. There you will understand this thing. Okay. So which we're going to be doing tomorrow, not today. Don't worry. Okay. So today we have to just get the facts and understood the importance of the things. So Char-gaffs rules say that purine will always make a bond with a pyrimidine or vice versa. There never be like purine making bond or pyrimidine making bond. Never gonna happen. Then it says that what ratio, if you have to find it out, you can find it out that how many purine is there, how many pyrimidine is there, how many this is there. You can find out in a DNA sample. Okay, and that's the very important thing. So what is that? Char-gaffs' first point was purine and pyrimidine are always in equal amount in a DNA. Equal amount in a DNA, because they will make bonds. So counterpart will be present in them, right? So that is what A. Okay. So we say that purine will be equals to pyrimidine. So we have what? Purine, if we have to say that A plus G, these are all the purine, will be equals to T plus C. They will be equal to it. Okay. Now, if you put A plus G upon C plus T, their value will be what? Their value would be 0. Okay. So purine and pyrimidine. So second thing is what? That A plus G upon T plus C value will always be equals to 1. Okay, because the amount is equals to 1. And okay, third point we're gonna be understanding. Third point is that A amount will equals to T and G amounts will equals to C. okay, will always be there. But if you will say that A plus T upon is equals to G plus C, no, that not gonna happen. Their amount would be equal, but if you put the ratios of these two things, it might not be. Okay, so this is very important. Means fourth point, if I write A plus T upon G plus C, it may varies from species to species, and that is what it is, a specific number that is present, species to species. Okay, but it's constant for one species. Okay, that's it. And this help in telling the genetic history or the genetic codes, okay, arrangements for it. Okay, so we have studied what? We have studied about the Char-gaff's rule, and we will imply it and understand it much better. Well, after this, the next thing or the last thing for today that we're gonna be doing is that how the DNA is and what are the parts of it. So I'm gonna be taking one more picture of DNA, and then we wanna be learning about it. Okay, so let's just take a good DNA photo, which I'm not getting. Okay, this will work. Okay, image is also not good. Let's take this. Now, if you see that, we're going to be taking what? We're going to be seeing it, what are the different things presented to it. So if you see over here, we have what? This is a B-type DNA, which Watson and Crick has seen it. Let's just see that what is presented to it quickly over there. So this is what, there will be opposite polarity present over there, right? They are what? Spirals. There is some major groups and there is some minor groups present to it. Okay, these are what? These are the cuts or depressions which are present into it. Okay, now you have to remember one thing, that this part which you see, this area, this is representing what? It is representing the sugar and the phosphate link. So that's why it is called a sugar-phosphate backbone. And on it is what? This nucleotides bases are present. Can you see that AT, they are making bonds. So these are what? On the backbone, there will be formation of what? Inside, which is projected. Okay. Now, if you see over here, this space is constant. Okay. And its value is 20 Armstrong. Now the question is, how it is constant? How it is constant? How that there is no, like somewhere less space, more space, why it is not there? So the answer to this question is that since it's always a purine is making bond with a pyrimidine, means the space will remain always equal. Means there will be what? If you see over here, beta, what is happening? See, this is the backbone, this area. If I have to just remove it, this area is what? This is the backbone. Okay, we have to see just this part. If you see here, how many rings you are seeing it? 1 2 3, 1 2 3. So always there are 3 rings that is present and making it what? At an equal size. Nowhere it is big or less. Of course, this has to be made like this. That's why. Okay, I saw it. So this thing is there. So if someone will ask you, what you will write? You will say that due to presence of purine pyrimidine bonding containing throughout 3 cycles of nitrogenous base compound equally gives equal measurement throughout. Measurement throughout. That's it. Now the next thing is that this thing, can you see? This is what, this from here to here, this is called as 1 turn. Okay, and the value of 1 turn is what it is, 3.4 or 34 Armstrong. This 1 turn is also called as 1 pitch. Okay, so if I have to write it up, so I'm going to be first marking it up. Wait, so from here to here, this is what, okay, they have just, from here to here, this is representing what? This is representing 1 turn. This is what? 1 turn. This 1 turn is also called as pitch. This pitch value is what? 34 Armstrong. Okay, and when it is 34 Armstrong, it contains 10 base pairs. 10 base pairs means what? 20 nucleotide. I have told you in the initiative of it, that is means 20 nucleotides. So if you see over here, we have what? We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So of course this is, diagram is not very proper, but this is like this. So we understood this thing. Now the next thing is, what is the distance between the two nucleotides? Like this 2 bases, what is the distance present? So this is what? It is this is 3.4 Armstrong. This is 3.4 Armstrong. That is what? Distance or space between 2 base pairs. Understood? So this thing is there. Now somewhere they might give you that instead of Armstrong, they will ask you in the nanometer. So Armstrong is 1 less, like 1 nanometer is equals to 10 Armstrong. So I hope you can change this thing now. Okay, so that would be what? This will become 0.34 nanometer. Okay, and this thing will become 3.4 nanometer. Okay, that's it. Okay, so these things you have to remember about the DNA, beta type of DNA. What are the other types of DNA? We have A DNA, B DNA, C DNA, D DNA, and Z DNA. Okay, so here only I am writing it, types of DNA. Types of DNA. We have A DNA. It has how many? 11 base pairs. Just remember it. Okay, then it is right hand coiling. Right hand coiling means when they are coiled like this. Okay, then we have what? Beta DNA, which we have already studied. How many base pair was there in 1 turn? That is 10 base pair, right hand coiling. Then C type DNA, we have 9.33 base pair. It doesn't mean that how come half base pair is there, because the average we take it out, it comes out that little bit is here, little bit in the next. Okay, this is also right hand coiling. Then D DNA, it is what? 8 base pair present, right hand coiling. And then we have Z DNA. It has 12 base pair present and it has what? Left hand coiling. Well, we have already covered a lot of things in this video. Please try to go back and revise what we have studied. These are the basic things which we have to do. Tomorrow we're going to be doing some of the numericals based on what we have studied today. So come back and don't leave that topic. Meanwhile, in tomorrow's topics, we will move forward that how this DNA is present inside our cell. Okay, different terminologies which you might have studied till now and had a confusion, we will try to make it sorted. If you like this video, give it a like, share it with your friends, because sharing is caring. It motivates me a lot. If you will subscribe me also. These notes would be available on my WhatsApp channel and WhatsApp page or channel, whatever it is called as, or the Telegram channel. You can get the links in the descriptions for both of them. That's all for this video. Thank you.